Okay, so just give me a minute. Okay, so hello, good evening, and welcome to the next session for this course. So, as you might remember, we were discussing the topics for genes, genomes, and the epigenomes, where we wanted to complete our discussion on DNA protein interactions, the basic concepts regarding the same, the techniques that can be used to study them. And under this entire subtopic, we had covered the domain on in vitro DNA protein interactions. And today we are going to move forward with in vivo methods, specifically focusing on chromatin immunoprecipitation and the logic behind it. And uh, we'll just introduce how chip sequencing is done, and I will go into more details about ChIP-seq in the next class. Today is more of an introduction to ChIP. In the next class, we'll discuss how the library preparation is done, what are the parameters to be kept in mind when you are sending it for sequencing, what are the tools that you need to analyze, and what are the sort of outputs you get uh, upon analyzing ChIP-seq data. And the reason why we'll go into it is because a lot of this information is already publicly available. So you should have an idea as to how you should make use of publicly available resources for your hypothesis, and then do the testing of the same hypothesis using whatever model system you are particularly interested in studying. Then we'll move on to our discussion on chromatin accessibility profiling using a DAC-seq, high-seq, chromatin conformation-based methods, 3C, and then we'll end the course with some of the few leftover topics that we have discussed beforehand as well. So, with this introduction in mind, we'll move forward. And first, we'll do a quick recap of what we had discussed in the last class. So, we had revisited the entire concept of DNA protein interactions and why they're important. Of course, it is an interaction that is central to the functioning of any cell. When I say functioning of any cell, it is in context of regulating the expression of a number of genes, which are eventually controlling the proliferation, survival, and sustenance of any cell. So, one major group of such interactions are or are facilitated between DNA binding proteins, which are classified as transcription factors. And among these transcription factors, we took one sort of example. Uh, for example, if you talk about any RNA polymerase for that matter, that is also a DNA binding protein. It is recruited at a specific site. Then we discussed about restriction modification enzymes, which are able to recognize specific DNA sequences, bind to them, cut within them, or cut outside the recognition sequence. But effectively, the interaction between these two moieties is what drives that process. So, studying these interactions, characterizing them, and understanding what practical strategies are applicable in this context, all of that falls under the purview of this particular topic. So, also, another additional factor to be kept in mind is that DNA protein interactions, particularly, are very relevant whenever you talk about developmental processes or you talk about sensitivity or to environmental stimuli. When we say environmental stimuli, we mean in the sense that if there is a change in the surroundings. For example, if there is a stressful environment and the cell has some stress sensors present, then the stress sensor should be able to convey that um, not even information, convey that stimulus inside the cell, and then the cell should be able to make a judgment as to whether or not it should continue with the processes it was continuing before, or it needs to shut down something, it needs to begin something. All of that needs to be taken care of. So, yeah, so basically, there are two entities that are obviously making this interaction possible. The first is the DNA moiety, and the second is the protein which binds to that DNA moiety. And the protein's identity, its molecular weight, its domains. Naturally, any DNA binding protein should have a DNA binding domain, DBD, and it should have another sort of activation or a response domain basically, which is able to execute its job apart from just DNA binding. So these are the two common entities, and the DNA sequence should doesn't necessarily need to have any specific properties, but basically it just needs to be accessible to the protein for it to be able to bind, to be able to um, sort of dock onto the DNA sequence, DNA motif, and then execute its activity eventually. So that is the background. Now we had begun our discussion with some in vitro techniques, an example of which was the footprinting assay. And as we have already completed this discussion, I'll go over it a little bit faster. But basically, footprinting assay is an indication of any region which is inaccessible to a restriction enzyme, which protects or shields some regions of the DNA from the action of restriction enzymes. And that leaves behind a footprint. Why? Because in the absence of that protein, we expect a complete smear of DNA of different molecular weights or different sizes, and any region which is bound by DNA is going to be protected, and that region will not be digested, or that fragments of that length will not be digested, and as a result, you will get a footprint of the protein binding and protecting that region at that point of time. So it involved the use of some chemical or enzymatic digestion of naked DNA and protein-bound DNA oligomers. Then they are compared using gel electrophoresis. A major factor that should always be considered in this context is that your DNA has to be labeled in some way or another. Otherwise, detection and visualization would not be possible. So most commonly or most primitively, what used to be done was that there was radio labeling, which was done for the 5' end of the DNA, which is getting sequenced. So commonly you would use a heavy isotope of phosphate so that you can exchange the regular cold 5' phosphate with a hot 5' phosphate, which is radioactively labeled. In today's time, you can opt for a fluorescent probe as well, but many a times this is still performed in a large number of labs. However, as much as possible, radioactivity should be avoided if you have a better option, and in today's time, we actually do. So once this is done, once labeling of the DNA is done, most commonly at the 5' end. Why at the 5' end? Because that will indicate how long the fragment of DNA is. Alternatively, 3' labeling can also be done, but it is going to, it can possibly lead to the exclusion of a large number of signals as well. So in case the DNA molecules that have been partially synthesized or which have gotten nicked or sort of gotten broken down, you would preferably not want to do that. You wouldn't want that to get labeled, but it can be done from both ends, but most practically 5' end labeling is performed. Then plus and minus the protein, you have to use both your possibilities. You can separately add your protein of interest after purification, and here purification is very crucial because if the protein that you're adding is from a cell extract, then it is possible that there is some sort of non-specific binding and the results you're getting are not reliable and not specific to your protein of interest. So then when you do DNase 1 partial digestion, again, this is something we had already discussed. Why partial digestion? Because you don't want complete chewing up. You want single stranded nicks to be generated, and then you will get your DNase 1 footprint from this digested link. So this is your example of a sequencing ladder, the control reaction, and these are your two footprinting reactions. This is where you expect your protein to be present, and that exact sequence can also be detected based on the length which you're getting. So this is one way in which it can be done. So footprinting has actually been a very valuable tool for sequence specificity for the dissociation constants of a large number of ligands that bind to DNA. most commonly, what is done is that you use an agent called as the probe. Smaller the probe, higher is the resolution that is provided, but its chance of cleaving DNA under the bound protein also increases. Now, enzymatic digestion methods, they include the use of DNase 1, MNase, MPE, which is methidium propyl EDTA iron, Fe2, copper phenanthroline, uranyl photocleavage, hydroxyl radicals, and iron complexes. Now, comparisons between different types of probes used for footprinting provide useful information on their relative merits and demerits. So, DNase 1 footprinting most commonly is used. It's a double-strand specific endonuclease which binds to the DNA minor groove and cuts the phosphodiester backbone. It was developed by Gallus and Smith for visualizing the binding of lac repressor to its operator sequence. The footprinting technique, it employs the use of single-end radiolabeled synthetic or natural DNA fragment, which can be included with your crude or purified protein sample under appropriate binding conditions, allowing protein binding. This fragment is then subjected to DNase 1 treatment with varying concentrations and then run on a denaturing page, processed and imaged. Now, where are the what are the bottlenecks in this particular experiment? First bottleneck is that DNase 1 should not be limited by any additional factors. What do we mean by additional factors? Like, for example, steric hindrance should not become a liability for DNase 1. So DNase 1 hypersensitive sites are preferred. What do we mean by that? Basically, these are sites which are not heterochromatinized or which are not part of nucleosomal DNA and they are exposed. So this is a nucleosome-free enhancer region which is accessible to DNase 1 digestion. Nucleosome-free promoter region, if it is present, that is also digested. There is a nucleosomal repositioning region as well. And if you remember, we had talked about nucleosomal breathing, that this is your histone, this is the bound DNA. Now, some amount of shifting occurs or some amount of loosening does occur between this DNA-protein interaction and the regions which get exposed as a consequence of nucleosomal breathing, they can also be a part of they can also be subjected to DNase 1 accessibility and as a result to DNase 1 digestion. So these are the possible sites, and any region which is heterochromatinized and so tightly packaged actually does not get exposed and is not digested by DNase 1. Okay. Sorry. Just a second. Yeah. So next, the DNase 1 partial digestion is something that comes into question. The idea of partial digestion means that instead of random chopping and multiple fragments being generated within each DNA fragment, you want a single nick, one cut per molecule is the principle you wish to follow. And that is done by titering out the abundance of this particular enzyme. So you limit it in such a manner that only partial digestion is facilitated, which creates a range of fragments that you saw we could visualize on the denaturing page. Now, upon examining a variety of experimental footprinting gels, it appears that if all sequences are cleaved without any specificity by DNase 1, then you would have similar band intensity. You would have same types of bands or same intensity bands present throughout the lane. But DNase 1 has partial sequence specificity, which means that some sites are hypersensitive and they show a more intense band compared to others. Second, end labeling of DNA is also very important. In a single reaction, it can cut both strands, which can lead to a mixture of positive and negative strand fragments, which are then separated on the denaturing gel. But because you wish to know which of the two antiparallel strands the protein under consideration does bind, then in a 5' labeled reaction, only this information is provided. Secondly, it's common to digest both these strands in separate tubes and then run them alongside each other. Lastly, there are a number of methods to visualize or analyze the final footprinting image, which ranges from visual inspection to creating a differential cleavage plot on the basis of a densitometric analysis. Finally, there are techniques which are available for quantitative analysis of binding affinity. Then you have bovine pancreatic deoxyribonuclease 1 or DNase 1, which has been derived from a bovine source. So this is an endonuclease which can work on both single-stranded and double-stranded DNA. And as a result, it can produce primarily 5' phosphate dinucleotides and 5' phosphate oligonucleotides. These are 5' phosphate dinucleotides and oligonucleotides that can potentially be produced. Now, the way in which DNase 1 works, that can also vary in a sequence non-specific manner. So the enzyme, it requires divalent cations, which are both calcium, magnesium, or manganese as cofactors for its full double-strand cutting activity. In the presence of Mg2 plus alone, when only magnesium is present, DNase1 is going to exhibit a nicking activity, and single-stranded nucleolytic activity would be more discriminatory. So you want to limit the same when you are carrying out your digestion for your analysis, for your footprinting assay. Thirdly, you have to run the sample on a denaturing page, and only single-stranded DNA would show up. And as a result, whatever is labeled with your at the 5' end with your radio label or your fluorescent label will be visualizable at that point. And it's chemically sequenced many a times and then run on the gel so that you can get an exact idea of what is the sequence that you are getting from your gel. So because that's going to narrow down another step and make your results a lot more trustworthy. And some precautions include that you do not titrate too much DNA with the protein sample. As a result, a large amount of DNA will be unbound and susceptible to DNase1 attack, which will show up instead of the footprint. That will make it much harder to detect. Second, incubating the crude sample with the fragment. If you are proceeding with that, then you need to have some sort of competitor DNA so that non-specific protein footprint can be excluded from that. So that is your control experiment if you're using a crude extract. That's why I mentioned it's always a more sound practice and a more reliable practice to have purified, just pure protein that you add in your in vitro experiment to avoid any such unreliable conclusions. Then other probes for nicking DNA, like free radicals, may not get hindered very easily, and therefore there can be multiple nicks which can be generated under the bound protein itself. Now, apart from this method, which is, while not primitive, but a little bit backdated, apart from that, footprinting is also used to elucidate the binding of other small molecules like drugs to DNA. Technique that uses DNase1 digestion followed by sequencing is called as DNase-seq, which is used for a number of genome-wide studies. Again, in today's time, you have a lot of not progressive, but a lot more advanced strategies for understanding the genome. So this is not really that relevant. And protocols using automated infrared sequencers that allow long range and highly sensitive footprinting have also been developed. Also, using streptavidin-bound oligonucleotides for protein binding and subsequent DNA digestion makes the technique also very convenient. It can also be used to fish out a protein of interest from a crude sample. It can be used for quantitatively measuring the dissociation constant of the DNA-protein interaction. So, sorry. Then we talked about EMSA, which is electrophoretic mobility shift assay, which is another in vitro method. Basically, when there is a positive interaction or when there is an interaction between a DNA molecule and a protein, you expect a shift in the electrophoretic mobility of the bound complex compared to the control sample. And that is what you visualize in a gel. So Yeah, it's based on the principle that these complexes are heavier, and as a result, they face more retardation and move slowly when they are subjected to a non-denaturing polyacrylamide gel as compared to the unbound free probe. And since the rate of DNA migration does get shifted or retarded when bound to your protein, the assay is often also referred to as gel shift or gel retardation assay. And then the DNA sequence is provided externally and incubated to the crude cell protein lysate. So the example of the EMSA experiment. Now, to perform the assay, you have your fragment of DNA containing the suspected binding site, which is labeled by P32, like I already mentioned, using T4 or T7 polynucleotide kinase. Um, if the transcription factor binds, then a new band will get visualized by autoradiography. Like you can see that when there is a gel shift, it indicates that there has been DNA protein binding. And that is basically what you see here, DNA plus protein bound complex. This is your bound complex. This is your free probe, which is not obviously not bound. This is another example where multiple mutants have been utilized for your to compete with your wild type sequence, so that you can identify the relevance of um, each individual base within that sequence, and to find out which is the exact motif that is necessary for binding of the protein to the DNA molecule that is under study. And these techniques have been immense contributors in early studies, in early research. If you study from the book called I can't remember the name, but it is by Robert Weaver, and it talks about the techniques or methods that have been used in cell and molecular biology, then you'll see that how the discovery of the entire process of transcription in prokaryotic and then in eukaryotic systems was carried out, and how immense fundamental questions have been answered. For example, the Tata box, or the RNA polymerase binding site, or the splicing site. All of these information have been identified using such beautiful techniques, such simple to the ears, but having very far-reaching consequences. These sort of techniques have been utilized. So the impact is immense, and we should, give credit where due. Uh, Anyway, moving forward a little bit faster, the most another protocol which or another step which refines the protocol a little bit, which sort of refines the technique a little bit further, is the use of an antibody as well. So by incubating the antibody along with your DNA protein sample, followed by gel separation, this complex can also be visualized as a super shifted band. And competition assays may be performed using unlabeled specific and non-specific oligocomplexes or oligoduplexes. Um, EMSA may be used qualitatively to identify sequence-specific DNA-binding proteins in crude lysates, and along with mutagenesis to identify important binding sequences within a given gene, for measuring thermodynamic and kinetic parameters, etc. Um, yeah, basically it is quite useful. Then we had briefly discussed PLA, proximity ligation assay, again, another strategy for protein-protein interaction more commonly, but also used for protein-DNA interaction. And because we didn't go much into detail about this, I will move past and we will begin our discussion for today. So before I get into the details of the topic, first thing I want to stress upon is that there is a lot of difference between the strategies used for in vitro and in vivo techniques. And we had discussed this in the previous class, but just to reiterate it, when we talk about in vivo, we mean that these interactions are occurring within the cell without us having to provide any pure protein, any pure DNA. It's not something that we as scientists are doing outside of the cell. No. Instead, what we are capturing is what is occurring inside the cell under its regular physiological condition and whether or not that interaction is present. So let's take one example. Why somebody might ask, like, what is the difference? So the difference comes in the fact that when you are just taking A and B, two objects, and you're checking if A and B interact with each other, they're sort of working like inanimate objects, right? However, when you are talking about an in vivo system, it means that you are capturing something that is occurring within the cell and is an interaction that is facilitated as a response to some stimulus. So, for example, if you are simply talking about a transcription factor binding to DNA, so if that is occurring within the cell at any point of time, then you can understand that this transcription factor has a role to play in expressing some gene, which is wherever it is binding, and therefore the expression of that gene is also relevant. You would have to compare that result with expression studies as well to make sure that whatever you're observing is um, well conclusive. But yeah, you can certainly do that. And apart from that, also, when you are doing an in vivo study, you can vary parameters and you can check what sort of a response occurs in response to different parameters. For example, you may carry out, for example, a drug treatment, and then you might check what is happening inside the cell, and then you'll see difference between these two conditions. So, several in vivo techniques have been developed to characterize DNA protein interactions. And the question arises, as I've already discussed with you, that the requirement of an in vivo technique much largely is contributed by the fact that there are limits to in vitro experiments. That is because within the cell, the way DNA exists and the way the protein exists, they are not similar to what you would expect if you add these two inanimate objects into a tube at the same time. So DNA inside the cell exists in a compact chromatin state that has very distinct properties from naked DNA. And usually in vitro techniques are not able to um, simulate the condition that is actually expected to be present inside the cell. So therefore, chromatin immunoprecipitation is a very powerful technique for exploring the role of the epigenome in an environmental exposure mediated disease and susceptibility. Briefly, it is known as CHIP, chromatin immunoprecipitation. It is a technique that investigates the interaction of a specific protein or its modified forms. Now, this is something which I've written very briefly because we are not it's not something that I can very easily cover. But basically, what I would want you to understand by this word modified is that most commonly, what you would expect is that there are certain markers, certain modifications on proteins which indicate certain conditions. Like, for example, we have discussed the epigenome previously, right? So each of these histones have certain residues which undergo modifications. Now, the type of histone modification and the residue on which that modification has taken place or has been um, put upon, that can dictate the effect of the presence of such a modification. So basically, what I mean by that is that certain histone marks are considered to be activating, some of them are called sometimes considered to be repressive, and so on and so forth. So there are variations in this context as well. And if you specifically want to capture, say, for example, active chromatin, then in that state, it is a good idea to capture histone modification, which is activating in nature, and then study the same. So DNA protein interactions, they play a very significant role in various cellular processes like replication, transcription, DNA damage repair, genome stability, gene regulation, and segregation at mitosis. So as the name chromatin immunoprecipitation suggests, this method utilizes chromatin preparation. What do we mean by chromatin preparation? Why are we not just saying DNA preparation? So this is another major distinguishing point from in vitro techniques, that in vitro techniques we provide naked DNA and we provide our protein sequence of interest. So like I said, two inanimate objects and you're just checking if they bind to each other. But when we talk about chip is actually capturing whatever state the DNA is in. So if a particular protein is binding to your sample at any point of time, then that means that area has been opened up, or that DNA is exposed, available, and therefore the binding of your protein is an event which is crucial enough for the cell to invest energy, to invest ATP, open up the chromatin, recruit chromatin remodelers, open up the chromatin, and then allow your protein of interest to bind there. So it is not something, it's not a decision made by the cell in a whim or in a fancy, but instead it's a very properly regulated procedure. So uh, yeah, so this is what we mean by chromatin capture. So it captures the chromatin in its nascent or its physiological state, irrespective of whether it is bound or unbound. That's how you capture it. And it utilizes chromatin preparation from cells to selectively immunoprecipitate the protein of interest and to identify what DNA sequence is actually bound to it. So CHIP has been extensively used to detect transcription factors, variants of histone protein, chromatin modifying enzymes, post-translational histone modifications, etc. So this is how CHIP actually works. Then Then we learn a little bit more about the technique. So David Gilmore and John T.L., these are two scientists who are known as the pioneers of this particular method. So what did they do? They actually determined the in vivo distribution of RNA polymerase in bacterial genes by covalently cross-linking the protein to DNA in bacterial cells using UV irradiation. So initial studies of cross-linking were done simply by UV radiation, and it is not just limited to this technique, but in general for cross-linking, one of the agents which was used was UV. I think in southern blotting as well to immobilize your DNA sequences onto a supporting membrane. UV was used, followed by baking at hot temperatures so that the DNA would sort of adhere or sort of get immobilized onto your membrane. So, uh, yeah, so UV cross-linking is something that has been done for a while, but now there have been modifications to this protocol, which has been standardized and is found to work pretty well. And we will discuss it also. So, yeah, so they performed UV, or they carried out UV radiation followed by cell lysis and immunoprecipitation of the bacterial RNA polymerase. And finally, the DNA that is attached to the RNA polymerase in the precipitate can also be purified and assayed by hybridization using probes, which correspond to different regions of known genes. So basically, to see where active RNA poly2 recruitment was occurring to find active sites of transcription. Later, they applied the same method to study the association of eukaryotic RNA polymerase II on the Drosophila Hitchcock gene, and the method was further modified by Solomon and Warshawski, and they used formaldehyde as the cross-linking agent instead of UV radiation. And sorry, just a minute. Yeah. Then got modified further, and they started bringing formaldehyde as the cross-linker. Another reason why using UV is not a very good idea is because UV is also a mutagen. So it can also change the DNA sequence to some extent. And it's, of course, not safe. And formaldehyde is just more reliable, a much better cross-linker. And yeah, they monitor the interaction between the Drosophila HSP70 genes with histone H4 and RNA polymerase 2. Now, what are the key steps or the key, yeah, let's call it steps in the protocol for CHIP or in the CHIP method? So first, it involves stabilization of the genome and the protein's interaction, or the DNA protein interaction undergoes stabilization. So what do we mean by stabilization? It basically means that because cells are dynamic and you are going to subject cells to a lot of different procedures, a lot of different steps. So it is often considered a good idea to cross-link so as to sort of fix whatever DNA protein interactions are present in your cell at the time that you have taken your cell out. So basically, you take out your cells, you make sure that, yeah, they look, appear healthy. You take them out, and then you sort of, um, how do I put it? Take them out, and then you uh, fix whatever interactions are present in that cell at that point of time. By fixation, I don't mean permeabilization at this point. I simply mean that you want to freeze the interactions within the cell and not allow it to undergo any further dynamic changes. So one of the reagents which facilitates this is actually going to be formaldehyde. So it stabilizes these genome protein interactions, and following that, you perform cell lysis, nuclear material extraction, fragmentation of chromatin material, pre-clearing of the solution to reduce non-specific interactions, and or non-specific precipitation, and then immunoprecipitation, elution of your precipitated DNA protein complex, and finally the reversal of stabilized protein genome interaction and characterization of DNA and protein. So this is the DNA protein cross-linking. Yeah. So after cross-linking using formaldehyde, let me just switch off my video. Yeah. After cross-linking of the DNA protein interaction, next you perform cell lysis. And after the cell is lysed, what you basically want is for all the components to come out. During this step, it is very crucial that your lysis buffer has inhibitors for DNAs. Otherwise, it's going to chew up your DNA. But it DNAs as well as proteinase, so that the DNA protein interaction remains stable. Then you have your sonication or enzyme digestion, followed by formation of fragmented chromatin, immunoprecipitation with your favorite antibody, your desired antibody. So we'll go over these steps, don't worry. So basically, sonication is going to break your DNA or shear your DNA. That's the correct terminology commonly used, which is going to result in smaller fragments of chromatin, followed by adding your antibody of interest, shown here in black, these Y letters basically that you can see. They go and bind to your DNA protein complex. Then you purify the DNA. You will basically get whatever DNA is present. The protein here would be digested. You would get your DNA sequence. You can either use that for PCR, chip qPCR, microarray, or sequencing, and you can analyze what was that region of DNA that was bound to your protein. Next, we will go over each of these steps in a little bit more detail. So the first step that I already talked about a little bit before, which is also known as cross-linking. So naturally, what exactly happens here? You are cross-linking or freezing the DNA protein interactions which are present within your cell at any point of time. So commonly these interactions are covalently stabilized, and usually formaldehyde is used as the cross-linking agent, like I already mentioned. Other cross-linkers might also be used. Um, Namely, you have DSG, which is disuccinimidyl glutarate, ethylene glycol, bis-succinimidyl succinate, dimethyl adipamidate dihydrochloride, or DMA, subetic acid, bis-N-hydroxysuccinimide ester, DSS. DSS is the most commonly used. And there's also a variation of the same. I think it's known as S3. These are all commercial variants. Um, and they can all be used depending upon what has worked best for your currently existing protocol. So formaldehyde-based cross-linking, while very useful, it does have certain limitations. It does not, unfortunately, maintain all, all types of protein DNA interactions. For example, co-activator interactions, transcription factors in case of hyperdynamic equilibrium. So it does not maintain everything. And other limitations of using formaldehyde is that it is a zero-length cross-linker. So it does not allow for much flexibility. If your protein complex is quite large, then the physical dimensions of formaldehyde is too small for it to be able to freeze interactions which are a little bit further apart because of of the large complex formation. So in that case, if you have larger protein complexes with complex quaternary structure, longer crosslinkers like EGS or DSG, they may be used. And also sometimes two-step cross-linking is employed for more sort of robust and highly efficient cross-linking. And the condition and timing for cross-linking should be optimized, and it can also affect the sonication process and also may be responsible for epitope sequestering. So what do we mean by that? Let's go over the image for better understanding. The first subtype, which we call chemical cross-linking, you have your polymer 1 and polymer 2. In this case, these are oligomers or DNA sequences basically. And you have a cross-linker between these two entities, which will be covalently bonded. The second possibility is that your cross-linker is present, but for initial cross-linking, then partial cross-linker might even get removed. Only a small entity might be present, and it is going to lead to covalent linkage between these two. Next, you have physical cross-linking. Now, while that is done, it is not very stable because it's a non-covalent interaction or non-covalent bond which is formed between the two. Then you have enzymatic cross-linking. So certain enzymes might also be used to facilitate the cross-linking between these two entities, and covalent linkages may be formed between the same. However, the most common subgroup which is utilized among all of these are the chemical cross-linkers, um, which facilitate interactions between these two proteins. The second step, after your first step being cross-linking, the second step is slicing your cell. So what do we mean by slicing the cell? You basically want the cell, you basically want to pull out all the DNA protein interactions from your cells, and for that you have to lyse, and you will bring these complexes into the solution. So naturally, whenever we talk about cell lysis, the sort of reagent which we would need is a detergent, and you would need some buffers. So generally, there's a chip lysis buffer, which is some of it is commercially available, some of it is usually manually made fresh whenever you're performing the experiment, because it's not something that is routinely done. It is done to answer a sort of question that is necessary for your study. So generally, treatment with chip lysis buffer is used for the lysis, and removal of the cytosolic part is also done as DNA protein complexes are present in nuclear fractions. However, mechanical lysis of cells is not recommended for this step. Hmm. Next step is the shearing of DNA. So till now, what have we done? We have linked our DNA protein complexes, and we have sort of ruptured the cell, yeah, and brought out all of these complexes into solution into the lysis buffer. Now, within the lysis buffer, what should we do? Now, we should figure out where exactly this protein has bound to the DNA. Now, because DNA is a very rather large and complicated molecule, which would be very difficult to analyze and is also going to give you very limited information if you study it as it is. Therefore, a good idea is to do shearing of the DNA. So, what do we mean by that? So, basically, the extracted nuclear fraction or the nuclear component, it contains A, the unbound nuclear protein, B, your intact chromatin, and C, the cross-link complexes of protein and DNA. Now, from this pool, you wish to only study the interacting DNA and protein regions. Long genomic DNA, therefore, should be fragmented to allow us to selectively pull out these interacting complexes. And generally, for that, you need to perform fragmentation somewhere between the range of 200 to 1000 base pairs. Usually, it's 300 to 700, depends on how you have. Like it's actually your lab's protocol, what standardization has been done, and what works best for the types of cells that you use and for the questions that you wish to answer. So sometimes you might actually need to standardize that. Now, DNA shearing is usually achieved either by sonication or by enzymatic digestion with micrococcal nuclease. So, how is DNA shearing done? Usually, you would do either sonication. Sonication can be done also by two methods. I'll discuss it soon. Or it can be done using MNase digestion. Now, MNase digestion usually we do not do in today's time. It's a better practice to use a sonicator, preferably something like Covaris, which has a very fine control on the length or the fragmentation efficiency of the instrument, and it can generate fragments within the range that you have commanded it to proceed with. However, each of these processes has their own limitations and advantages. Enzymatic activity can be very crucial for shearing, and differences might lead to very variable results. Otherwise, DNA shearing is highly reproducible by enzymatic treatment. Sonication needs high optimization and the maintenance of a cold temperature. And the sonicated chromatin can be stored up to 3 months in liquid nitrogen at -80, and multiple freeze thaw cycles should be avoided. Size of the fragmented DNA can be determined in a 1 or 1.5% agarose gel electrophoresis protocol. Yeah. So this is what you see here, your complete DNA. Then you can perform a histone chip or a non-histone chip. So depending on that, your protein may or may not be bound here. If your experiment is a non-histone chip protocol, then it is going to be a protein which binds to the exposed region. If it is a histone chip, then it needs to be a part of the nucleosomal DNA. And yeah, then you can compare the two using different strategies, which we'll discuss. But if you look at the key steps first, you fix DNA and protein interaction, or you chemically freeze it using certain reagents. Second, you have to prepare your chromatin, you have to shear it, and the shearing allows for pull down of protein DNA complexes. The DNA protein pull down complex antibody and your protein histone modifications are what you have at the end. Now, Covaris through chip benefits. Only formaldehyde monomers allow for reproducible fixation. Um, Optimized buffers. Why I'm talking about Covaris is because if present, it is a very convenient way of sonicating fragments within a desired size. Optimized buffers are already present. Non-contact, random, isothermal, which means simply very close to the temperature, it maintains temperature. Focused, highly reproducible, shearing with epitope, and cross-link presentation. It can be paired with most commercial kits and homebrew methods. Very little chromatin dilution is actually required, 1:1. Better chromatin antibody ratios and better IP efficiency, especially for very limited samples. So these are the two common machines that I was talking about. The first one is called as a bioruptor. So it's usually produced by this company called Diagenode, which basically allows you to place your tubes containing the sample, which has your DNA protein complex or the extract basically into this rotor. It sort of has multiple cavities within which you can place it. Here you can program the shearing cycle, and then you can expect at the end of the program that there would be sheared chromatin, that it would consist of sheared chromatin of all of these different sizes. Um, Now, this is a sort of a representative image of what a gel would look like after this digestion procedure. So, compared to genomic DNA, if there is incomplete sharing, you will see a very high band, which is similar to what is expected from genomic DNA. However, with this proper sharing, you would get sort of like a smear throughout because it has fragments of all of these variable sizes. So, the other strategy for chromatin shearing is known as covariance. Covarice is a much more sophisticated instrument, which is based on acoustic methods. So basically, it involves a controlled environment with very focused acoustics that is able to using ultrasound waves that is able to cause DNA shearing, and it has a different mechanism of control, which is going to generate fragments within the range that you have commanded it to proceed with. So you need to provide the duty factor and you have to provide the intensity, cycles, and the time, and then it just runs as per your command. If there's a standardized protocol you can get from somewhere, you should try it out. And as an outcome, what we get from it, what we can actually use for our analysis is to check the peak for the percentage of or the size of the base pairs that have been eluted. So this shows you a very broad range where you have these fragments which are around 100 base pairs and some fragments which are around 10,000 base pairs. So there can be a very variable range which might be utilized for this purpose. Next step is the most crucial one. So till now we have cross-linked our DNA and our protein of interest. Then we have lysed the cells to bring these components out, and then we have performed immunoprecipitation. Now in this step, chip validated antibodies are to be used. Yeah, for the selection of target proteins, and then interactions and all the other protein components will be removed. Now, the antibody you do use in this case might be monoclonal, polyclonal, or oligoclonal specific for the target protein. If these are not available, then inclusion proteins, that is, target proteins may be tagged with his, MIC, human influenza, hemagglutinin, GSTT7, B5, etc., may be expressed in the biological samples, and then that can be used. Yeah. So antibodies, which are then specific to that particular tag, whether it is GST, B5, HA, etc., will be utilized for immunoprecipitation. After the precipitation reaction, complex of the antibody protein DNA is purified using beads that bind to primary antibodies used for immunoprecipitation, that is protein G, A, or protein AGVs. Then beads need to be blocked. What is the blocking of DNA? Basically, if you have studied Western blotting, this is a much more upgraded version of Western blotting. So you need to, whenever you use a measuring, you should always block it. So similar to that, here you have to perform some washers and then you can block it. And this blocking usually is performed using salmon sperm DNA, herring sperm DNA, and BSA, bovine serum albumin, which is used as a blocking agent. In general, there are many alternatives to detect an immunoprecipitated chromatin, such as PCR, qPCR, labeling and hybridization to genome-wide or tiling DNA microarrays, molecular cloning, sequencing, or direct high-throughput sequencing, which is ChIP-seq, which are all variations. So here as well, just to recap what we have already seen, cells are grown, they are cross-linked using formaldehyde. Then cells need to be lysed. The chromatin needs to, yeah, the cell is lysed, and then chromatin needs to be sheared. Then you add your specific protein targeting antibody. Then the DNA protein interactions can get precipitated by the antibody. So how does that work? Say, for example, if for protein X, there is an anti-X antibody, Yeah. So, protein-specific antibody can be added, then the DNA protein interactions can be precipitated using a secondary antibody, which is no, can be precipitated using the antibody itself. And yeah, then you can get DNA protein interactions, which can be precipitated by the antibody, and then specific interaction complexes are captured by the protein AGPs. Alternatively, these can be decross-linked at higher temperatures and then analyzed, and then qPCR sequencing microarray. After elution, qPCR or sequencing or microarray analysis might be performed. Now, what you do after this, you want to reverse your cross links and you want to characterize the DNA which was bound to your protein of interest. Now, that can be done using heat incubation, proteinase K digestion. However, if the DNA protein interaction is quite tough, high salt treatment might also be used before the proteinase treatment, and then DNA can get purified using PCI and using DNA purification with kits or columns. When you have DNA analysis, now the final output as the result of this experiment is DNA analysis. So most commonly, most affordable of these options is using a PCR chip qPCR method. If you have only a small scale, yeah, it can be done using multiple methods, including PCR, quantitative, real-time PCR or amplification for chip-on-chip, Sanger or cloning strategies. So that's what you see on the right as well. You have your DNA molecule that is getting cross-linked to the cells. Then after lysis, you have the elution of or the release of all of these cells. Then you lyse cells, allow the DNA to come into solution. Then you perform antibody binding. You add the antibody. You immunoprecipitate. You pull it down. Then you add a few washing steps. You reverse the cross link and you analyze DNA using quantitative PCR. Now I will end today's class using a few variations. I think we have, yeah, three variations only. And I'll be back in just 2 minutes. Yeah. Okay. Anyway, so we are next looking at some way. I think it's joining. Just give me a moment. Never mind. See if he is able to join later. Okay. So, yeah, I was talking about some variations or some changes in the or some alternatives to the common chip technique. The first one is X chip. So this method, it allows for the freezing of all DNA associated proteins by cross-linking using formaldehyde. So we've already discussed Okay, so joined a bit late. So just to catch you up, we are discussing DNA protein interaction methods, in vivo techniques which are used to study DNA protein interactions. So I have already covered the concept behind chromatin immunoprecipitation before we proceed with ChIP-seq. But basically, the idea is to capture the in vivo DNA protein interactions that are present under nascent conditions or under physiological conditions inside the cell. And you do that. One of the methods by which you can do that is chromatin immunoprecipitation. And one major advantage of using these techniques over the in vitro methods is the fact that you can capture the interaction of a protein or its modified forms as well. An example of the same is when you wish to capture histone marks, which are of different types. Either they can be repressive or they can be activating in nature. So, depending on the question you wish to answer, you can perform different types of chromatin immunoprecipitation using antibodies against these modified forms of the protein as well. In addition, you can also look at other sort of marks like RNA polymerase 2 in its phosphorylated state. You can use antibodies against that as well. Even there are some antibodies which are able to detect the number of modifications. For example, whether there is a single methyl group or a di or trimethyl modification which is present on your target protein, and you can detect that specific modifications chromatin immunoprecipitation mark as well. So the applications of this technique are limitless. So with this idea, we moved forward with the overall concept. So CHIP has these steps. Commonly, you have the first step involves cross-linking of your of the genomic DNA and your protein of interest. Most commonly, that is done in today's time using formaldehyde. Alternatives to that are also present. I'll come to that in the next 2 or 3 slides. The idea is that you basically want to freeze all of the modifications which are present in the cell under physiological conditions at that point of time. You do not want any dynamic changes to occur within the same. Following that, you will lyse the cell so that all of the DNA protein interactions which are present can come out into solution into your buffer that you are using at the time of lysis. And during lysis, one thing you must keep in mind is that you should have inhibitors for DNAs and proteinases so that they do not end up uh, digesting your target molecules, and that is going to completely skew your results. Following that, what you get is your lysate, which has all of these DNA protein interactions frozen or stabilized by use of the crosslinker. Following that, you should shear your DNA. Shearing basically means fragmenting your DNA so that you can capture smaller regions, or you can capture the exact region where binding has occurred. And also because many a times these long stretches might have multiple interactions, so you might miss out on some information, or you might have incorrect information that you might conclude from the same. So fragmentation is a crucial step, or sonication or fragmentation or shearing is a crucial step. Following which you perform the actual immunoprecipitation step. And this immunoprecipitation naturally means you use an antibody against your protein of interest, and you incubate that with this sheared chromatin. And any region where your epitope is exposed on your protein of interest, the antibody can bind. And consequently, the immune precipitate along with your protein of interest. What you will get in your pull down is also the bound DNA. Next, you have to purify that bound DNA because that is your target molecule of interest, and that you do by removing the protein, removing any RNA, etc. You decross-link your protein and DNA, and then you digest the protein and you get your pure DNA, and then that you can use for your analysis. Commonly, for analysis, either you can run a simple genomic a simple agarose gel basically to check what has happened after PCR, or you can quantify the amount of protein bound DNA using qPCR. This is what we most commonly do before proceeding for ChIP sequencing, if you're not proceeding for ChIP sequencing, or if you have analyzed ChIP-seq data and you wish to confirm whatever you have observed using your in silico analysis, if you wish to confirm that using your model, practically you want to validate it, then you perform chip qPCR if you're not opting for chip sequencing. And previously, microarray also used to be done. That also I'll explain very briefly. And of course, ChIP-seq is something that people naturally do. And it is obviously anything which involves high throughput sequencing is going to be much more expensive. In comparison, if you proceed with PCR or qPCR, you simply have to design primers and run a PCR. So that's going to cost way, way less, like 100 times less probably. So yeah, that was the overall idea. Then we discussed the different types of cross-linkers. These are all the different names of different chemical cross-linkers that can be commonly used. And while formaldehyde is the most commonly used reagent for the same, one limitation to its usage is the fact that because of the size of the molecule, because formaldehyde obviously is a very small molecule, it is not always able to maintain all types of DNA-protein interactions, which especially are which are the result of large ternary complexes forming a huge chromatin loop. In that case, if your DNA molecules are not very close together, if your protein and DNA molecules are not very close together, then formaldehyde might not be able to facilitate such a large cross-linking in the physical space just by virtue of its small size. So in that case, you have to opt for longer cross-linkers like EGS or DSG, which along with the dimensions that it is actually able to capture, has also been mentioned here. Sometimes you also can do two-step cross-linking for more robust cross-linking as well. And this is just an example of how this cross-linking occurs. So you have these two molecules. Here it has been represented as polymer 1 and polymer 2. In context of chromatin immunopresentation, it is a DNA molecule. These are two DNA molecules. And alternatively, you can also have a DNA and a protein molecule which are present, and you can cross-link those as well. So apart from that, you can do physical cross-linking also. Sometimes UV-based irradiation or UV irradiation is also used for cross-linking. However, that is a little more primitive. It used to be done in very initial studies which were carried out by scientists. And you can also do enzyme-based cross-linking, but most commonly chemical cross-linking is what we do in practice. Then once you have frozen those DNA-protein interactions in physical space, you perform cell lysis, where you basically permeabilize the nuclear membrane as the cell membrane and the nuclear membrane to bring out all of your DNA protein complexes into your buffer solution, into your lysis buffer, and you want to enrich for the DNA protein complex, and you remove the cytosolic part as well. Mechanical cell lysis is not recommended. One reason being that if you are too harsh with your cell in these circumstances, what you might end up doing is while it's not always something that happens, but it is definitely possible, is that you might end up disrupting DNA protein complexes if you're too harsh. So preferably you should go for chemical lysis, and you can scrape your cells to some extent. Then comes the shearing part that I just mentioned, the fragmentation of DNA or shearing of DNA. So usually the length which is chosen is somewhere between 200 base pairs to 1000 base pairs, or sometimes an even tighter range may be chosen, somewhere between 300 to 700 base pairs. And this shearing is facilitated most commonly by sonication based methods, or sometimes you might also use enzymes like micrococcal nuclease. Sonication is again the more preferred route because it is more reliable, but micrococcal nuclease or enzymatic digestion has more reproducibility in terms of results, because enzyme-based digestion will be at specific locations, but sonication is going to generate random breaks in your DNA molecules, and that is going to be a little less reproducible, but more commonly utilized once again. So DNA shearing using sonication needs optimization as well, and also needs to be maintained under very cold temperatures. And this sonicated DNA can also be stored and can be used for analysis later on as well. Now, for sonication, commonly two strategies are used, which I'll explain also in the next slide. So one is Covaris and the other one is the use of bioraptors. Basically, they are sonicators from two well-known, well-established companies. The first one which is known as Bioruptor, which is owned by a company known as Diagenode. They Here is what you see, something in black, a cylindrical sort of structure is a holder for your samples. So your lysed sample containing DNA protein interaction complexes would be placed within this holder. And here you can set the program via which you want your digestion to be carried out, shearing to be carried out, and then it carries out the same within the instrument. Another variation, a more sophisticated version of the same. This is the machine. It's a tiny compact machine. Here, like if I zoom in, here is your holder, and this entire block is able to maintain the temperature that I was talking about. That cold temperature needs to be maintained for sonication. And within this holder as well, I don't think it's going to zoom any further, but within this holder is where you place your sample. Now, keep in mind, while Covarius sounds amazing and is very, sophisticated, its reagents are more expensive. It's a more sophisticated instrument, but gives you fabulous results also. So you again place your lysate containing DNA protein complexes within this tiny little sort of tubes, glass tubes, and you place it inside. And here to with the attached system, you have the software installed, which is exclusively for this instrument. There you can set your program. So the computer control allows you to set these parameters, which includes the intensity, the duty factor, the cycles, and the time, which is going to basically set a program for how this shearing needs to be carried out. Again, there are commercially available protocols. There can be standard protocols. You can contact the company. Opportunities are endless. And what it basically does is it's going to, using focused acoustics, it is going to it is going to uh, introduce some sound waves basically to your sample, which is going to cleave it or shear it at the dimensions that you have specified to the instrument. Like I mentioned, either somewhere between 200 to 300 base pairs, up to 800 or 1000 base pairs. Again, this is controlled by the parameters that you have set for your instrument. So these are these are some commonly used strategies for DNA shearing. Now, what you get as a result of this shearing looks somewhat like this. If you run this entire sheared DNA onto a gel, it'll give you some output, something like this. So this is your unsheared DNA, which is very large, and obviously it is not able to migrate through the gel, versus very well sheared DNA is going to give you a complete smear. Don't get worried. Uh, this is actually ideal in this case because you want fragments of these desirable sizes. Actually, if you refer to your molecular weight ladder, you'll see this is somewhere 1 kb and this is around 500 base pairs. So most of your fragments should be located within sort of this range. And if you see some residual band somewhere close to the undigested, unsheared DNA, then that's going to represent that some of your DNA is incompletely shared or has not been shared and has stayed behind as contamination. But that is okay. Even if you have a good percentage, you can proceed with immunoprecipitation with that DNA. It's going to give you the results you wish to observe. And from Covarius, you also get an output somewhere like this, which shows you the approximate abundance of the length of fragments that you have gotten as a result of shearing. So somewhere between 200 to 12,000 base pairs. You can see that you can see different curves for different base pairs, and these are the ranges that you've shown. Again, this is depending on what program you have set for the instrument. Now, after you have sheared your DNA, the next step is to proceed with immunoprecipitation. Now, this I think I have previously discussed before as well, but the idea is that you basically use an antibody which is specific to your protein, or as I mentioned already, a modification on the protein that you wish to study, which is representing a certain cell state. And you incubate these together. And what you expect as an output in that case is that when your protein specific antibody is added to the DNA protein complexes which you have gotten. Again, keep in mind that not not every DNA molecule is going to have your protein of interest, right? So using this antibody is going to specifically pull down those DNA fragments which have your bound protein so that you can study the region where the protein was originally bound. And these DNA protein interactions can be precipitated, and specific complexes can be captured using protein AG beads, or more commonly used in chip experiments are dyna beads, which are magnetic beads because they provide even lesser contamination. Again, they're a little more expensive, but they do their job really well. Dyna beads or any other magnetic beads from any company which you're familiar with. And it also causes less mechanical stress, like for protein AG beads or protein cephalosporin beads. What happens is that you have to centrifuge every time you have performed an incubation to pellet down the beads and you remove any supernatant. However, if you use a magnetic beads, then the instead of having to centrifuge, you can use a compatible magnetic stand to which your magnetic bead is going to bind. And as a result, you don't have to spin down your lysate every time. And that reduces the stress and that reduces the possibility of disruption of DNA protein complexes every time. Because here you want these interactions to be present. And while you have added cross-linkers, you do not want to miss out on any signal. So it's just another precautionary move. So, yeah, so these DNA protein interactions can be precipitated using your antibody so that you can pull down your DNA of interest. So these antibodies that you use might be monoclonal, polyclonal, etc. Preferably, always you should go for monoclonal, and you should make sure that the epitope that it binds to is something that is exposed on the cell surface, and it is not present at the interface where the DNA and your protein of interest interact with each other, because that is going to compromise your results. And in case you are working with a protein for which you do not have an antibody, then most commonly you can opt for a tag specific antibody like HA tag, V5 tag, flag tag, etc., his tag, MIC tag, etc., which can be present attached to your protein of interest. And that usually happens if you have overexpressed your protein of interest in a vector. And then you can use the tag specific antibody to pull down samples, and that's going to give you even more pure results. And it is going to specifically detect only the overexpressed protein and not the nascent basal level protein expression. So, yeah, after the precipitation reaction, this complex can be purified, and you also, much similar to the protocol which is followed for Western blotting, you also need to perform something called as blocking of the beads. Basically, think of the beads like the parallel to that of a membrane, a PVDF or a nitrocellulose membrane that you use for Western blotting. So there, the membrane is where your proteins are binding. Much similar to that, here your beads are where your antibody is going to bind. So you do not want any non-specific binding. So for that, first you need to pre-clear the lysate. You need to remove any non-specific binding protein from the lysate, and you need to discard those beads. Second, you only wish for your specific antibody to definitely bind to to the bead, and that incubation is also should be allowed to occur for the required duration. Then you use wash buffers to get rid of any non-specific binding, and then you block it also to make sure that unbound regions are protected by some blocking agent like herring sperm DNA or salmon sperm DNA or BSA, bovine serum albumin, something of that sort. So what that is going to do is it is going to eliminate unnecessary background noise. Any non-specific antibody binding giving you false results is not something you desire. Because if you read a little bit more, you will find out that chip by itself is an expensive experiment because it requires quite large volumes of, when I say large, I mean in microliters, but still large because antibodies are expensive. And monoclonal antibodies particularly are quite expensive. So you would need quite a lot of antibody because these are, as you can understand, it's not like Western blot where you have an abundance of your protein, but instead you are trying to capture a very nascent interaction within a cell at a given point of time. So the abundance of your protein possibly is going to be quite limited, unless it's a sort of a housekeeping gene or something of that sort. So therefore, very concentrated amounts of proteins are required, and therefore the amount of antibody that goes is high. And so you have to be very careful with this experiment, and you want to minimize background noise as much as possible. So that's what happens there. And there are multiple alternatives also to immunoprecipitated chromatin to detect this immunoprecipitated chromatin, like there's PCR, like I mentioned, qPCR, chip on chip, ChIP-seq, etc. We'll discuss a few variations in the next couple of slides. Now, after immunoprecipitation and washing, you have to perform reversal of crosslinks and DNA characterization. So heat incubation, proteinase digestion, etc. needs to be performed. RNA digestion also should be performed for it, eliminating any non-specific results from that point. And then high salt, low salt purification needs to be done so as to salt out the DNA from solution. And sometimes also glycogen is added to enrich for DNA precipitation from the sample. So basically what has happened till now is that you have incubated your DNA protein complex with the bound antibody and incubated it with the bead till this step, which is immunoprecipitation. Once this is done overnight and once this has been completed, now what you wish to do is you want to get rid of everything else, the protein and all of these non-specific proteins, and you only want the DNA out of all of this. So to do that, first what you do, you get rid of this unbound DNA, you take all this bound DNA, wash it out, get this clean product, which is only your bead, antibody, protein, and DNA. This quaternary complex is what you want. From this quaternary complex, you want to get rid of 1, 2, 3, and you only want the DNA. So to do that, you have to remove the cross-linking. And all of these entities are proteins, so you can digest it using proteinase, and what remains behind is your DNA molecule. Now, this DNA needs to be purified, and the purification is done using high salt, low salt buffers, which will basically bring the DNA out of solution and form a pellet. Why? Because currently it is suspended in a wash buffer solution, right? So that is done using these buffers, like I mentioned, and using ethanol precipitation, using glycogen precipitation. This takes another 1 or 2 days. And once that has been done, what you get as the pellet needs to be resuspended in clean water, in molecular grade water. And this final product is your eluted DNA. This eluted DNA is what you have done all of this process for, very laborious, very elaborate process for. And this DNA is what you proceed with for your further experiments. So this DNA and protein analysis is done using the strategies that I mentioned. And yeah, so this is where we were when you joined the class. We talked about X. We were just starting to talk about X-chip, which is a sort of subtype of chromatin immunoprecipitation. So this allows us to freeze all the DNA-associated proteins by cross-linking using formaldehyde. Formaldehyde, like I mentioned, is a chemical cross-linker which reacts with primary amines, which are located on amino acids, and it also interacts with DNA RNA molecule bases, nucleotide bases, and forms a covalent cross-linking between the two. Now, these DNA protein complexes can be isolated by cell lysis, like we talked about, and the crude cell extracts can be sonicated, sheared the DNA to a smaller size. Then protein DNA complex is immunoprecipitated, reversed by heating. Proteins are removed by proteinase K. DNA portion of the complex can be purified and identified by PCR, and the use of formaldehyde for cross-linking minimizes nucleosomal rearrangements and is a very efficient method to analyze those proteins which have weak binding to your DNA. Now, like we already said, it is not very useful for capturing proteins which are quite uh, capturing protein DNA interactions, which are part of large complexes. So other bifunctional cross-linkers can be used, and the yield of chromatin and its resolution can be less after sonication, and there can also be epitope disruption. So these are all troubleshooting steps that you have to take care of. So this is just a representation of what it looks like, because mostly when we think about DNA, we think about naked DNA. Now, that naked DNA interaction is something which can occur only in vitro within a physiological system unless you deprive a cell of histone proteins, which is not something you would ideally do because that is not going to allow the cell to survive for a very long period of time. So under those circumstances, this is what the protein bound DNA actually looks like. And yeah, so then you fix cells with formaldehyde, you permeabilize and you sonicate. And this is XGypSeq. And after that, you get these little pieces of DNA. You use the antibody and then you immunoprecipitate, clean up the DNA, and you get this output. So this is the XGyp method. The second one is native chip or N-chip as it is sometimes called. It is a technique for natural DNA protein interactions where proteins are tightly associated to chromatin in their native state, such as histones. Like I already mentioned, histones that have a default high affinity for DNA and that do not require additional stabilization using cross-linking reagents like formaldehyde. And native chromatin within a cell can produce smaller fragments by treatment with micrococcal nuclease. An enzymatic digestion strategy instead of sonication may be used. And then immunoprecipitation can be performed using antibodies against your protein of interest. Now, enzymatic digestion technique is mild. It does not result in loss of antibody epitope during immunoprecipitation, yielding much higher immunoprecipitation efficiencies. And also you can get higher resolution because it can go up to single monosomes of 175 base pairs. Basically, it can, because of the enzymatic digestion, it can cut right after your H1 bound DNA, histone H1 bound DNA. And so you can get very tight fragments. However, the digestion can be uneven as enzymes can favor certain regions of the genome sequence, and as a result, you do not get you can get biased results. And to avoid such overrepresentation of sequences or overlooked data, comparison should be made with XGIP also. Additionally, nucleosomes can rearrange during digestion, and that also has to be taken care of if you're performing native ChIP-seq. So this is just an example of this method. Now, ChIP-seq is a long protocol. This single experiment takes up to 4 days, 4 to 4.5 days. And after that, you do PCR with this. So it's a complete 5-day protocol if done at one go, and very elaborate work at that also, but it gives you some very important data. So this is your cell lysis step, fragmentation using MNAs. This is your all part of chromatin precipitation, dialysis step overnight. Normally this does not need to be done, but for native chip, this is an additional step. Then you have chromatin IP, which is the immunoprecipitation step. Then you have library preparation and sequencing. Don't worry about this. I'm not going into detail because we'll discuss ChIP-seq protocol in the next class, uh, where I would start with library preparation itself, and we'll end with how ChIP-seq data actually looks like and what tools are necessary to interpret that data so that you can make use of publicly available data sets. So we'll move past that. We look at the next variation of chip, which is called as carrier chip. So this is based on immunoprecipitation from very few cells. If your sample is very, very limited, up to somewhere around 100 cells, and is suited for examining histone modifications associated with developmentally regulated genes, then this becomes relevant. So IP of such a small amount of chromatin is carrier chromatin from Drosophila or from any other species which is evolutionarily quite distant from the species which you are investigating, so that there is efficient precipitation of target chromatin. So here also you go for native chromatin digested with MNAs, and you use immunoprecipitation. For immunoprecipitation, you use antibodies to modified histones. And the low amount of chromatin can be detected by radioactive PCR, phosphorimaging. In today's time, you can go for quantitative dt-PCR as well, but it's not the most commonly used strategy, but it may be done. However, it requires very, very specific primers to be designed with very high specificity and to prevent any spurious amplification of carrier DNA instead of target chromatin. But basically, you are simply spiking your sample with more DNA so that immunoprecipitation can be facilitated. And within that population, you have to make sure that what eventually gets detected using PCR is only your desired DNA and not the carrier DNA. So primer designing becomes a very crucial step here. Next, we have chip, or as the name suggests, this first chip and the second chip are different. The first chip is chromatin immunoprecipitation. Second chip is a microarray chip. So this is a technique that combines chromatin immunoprecipitation with microarray technology. So there you label your immunoprecipitated fragments with a fluorescent dye like Sci5, Alexa 647, and then you combine it with genomic DNA that is labeled with Sci3, which becomes your reference DNA. So reference DNA is labeled with one dye. Your fragment, pull down fragment or immunoprecipitated fragment is labeled with another dye. Then this probe mixture is applied to the microarray chip, which consists of the whole genome, and you allow hybridization. Results of this experiment, what will it show? It will show you which regions of DNA are enriched by immunoprecipitation, because wherever it will bind, the fluorescent signal will be different, and then you will get a readout for your detected protein. Hence, the results signify the regions which are enriched, and hence chip data is obtained in the form of one-dimensional series of signals with peaks that identify that these are the regions that are bound by my protein of interest. Since the exact location of each arrayed element on your microarray chip is known, you can get a complete genome-wide map of DNA-protein interactions, which is somewhat similar to ChIP-seq, but it has certain limitations also. Now, while the chip technique, it offers several advantages over traditional chip assays, it is using it is giving you a comprehensive view of a large number of genomic regions. In fact, almost the entire genome is getting represented in one single experiment. It is eliminating bias, it is saving you time. Secondly, it is giving you the ability to study the localization of the protein binding, dismissing the need of running PCR assays as well. Third, it allows for parallel analysis of different genes to be classified in different classes, which is also good for their statistical comparisons. But despite this, microarray technology, if you have already studied it, you would know it has certain limitations. In today's time, if you want to get a comprehensive idea of your proteins binding, genome-wide binding in a cell under a certain condition, the best way to do it and to quantitatively also assay it is to go for chip sequencing. It is certainly not very cheap. It is pricey, but it is going to give you complete information that not only you, but scientists across the world can use once you have deposited it in the public domain. So this is something that we should keep in mind, and we should try to if we have the budget for it. And lastly, this I have not given the text for it, but just to give you an idea, many a times the sequential chip, or V-chip as it is sometimes called, is also performed when you wish to check for the co-occupancy, basically the co-occupancy of two different proteins in very close proximity within the same region or stretch of chromatin. So basically this is what it looks like. You have your proteins motif 1, motif 2, 3, so on and so forth, and you cross-link your chromatin, and then what you wish to see is, for example, if protein X and protein Y are part of the same ternary complex, which are localized very close to each other within the same stretch of chromatin, then you would be able to detect that. So first you perform chip with one antibody, say for example against protein 1. Then you perform chip with antibody number 2, which is going to give you an idea of two antibodies which are co-localizing on the same stretch or the same fragment of DNA. And that is going to complete, it is going to give you an idea that these two proteins are involved in some sort of a transcriptional complex, and they are able to carry out similar, they're able to control similar physiological processes. So then you uncross-link and then you can identify. So it's going to give you additional data regarding the same. So yeah, that is pretty much what I wanted to cover for today. So if you have any doubts, please let me know. And then we'll move on to our discussion for the next class, which will be on ChIP-seq and yeah, the analysis or the tools for studying ChIP-seq data. So any doubts, you can go ahead and ask. Otherwise, we'll end the class for today. Okay, if not, then I'll end the class for today. So thank you for joining. I'll see you in the next class. Good night.